Hello? 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 Morning. ಪ್ರಶಾಂತ್
Very good morning, sir.
ಗುಡ್ ಮಾರ್ನಿಂಗ್.
Very good morning, sir.
ಫೋಟೋ ಕಳಿಸಿ.
One minute, sir. Hello, sir.
ಹಲೋ, ನಾವು ಮಾತಾಡಿದ್ದು ಇದೆ. ಇಲ್ಲ, ನಮ್ದೇನು ರೂಮ್ ಪ್ರಾಬ್ಲಮ್ ಅಂತೀರಿ? ಅಲ್ಲ, ನಾನು ಆಪಲ್ ಅಂದ್ರೆ ಈ ಕಂಪ್ಯೂಟರ್ ಇಂದ ನನಗೆ ಅದೆಲ್ಲಾದ್ರೂ ಸೆಟ್ಟಿಂಗ್ಸ್ ಏನಾದ್ರೂ ಇದೆಯಾ? ನನ್ನ ಮುಟ್ ಮಾಡಿಬಿಡಿ. ನೀವು ಇದರಿಂದನೇ ಮಾತಾಡಿ.
Sir. Echo ಬರ್ತಾ ಇದೆ.
ಬೇರೊಬ್ರು ಮಾತಾಡಿದ್ರು ಕೂಡ ಈಕೋ ಬರ್ತಾ ಇದೆಯಲ್ಲ.
ಹಲೋ ಸರ್.
ಮಾತಾಡಿ.
ಹಲೋ ಸರ್. ಹಲೋ ಸರ್.
ಹಲೋ, ಮಾತಾಡಿದ್ರು ಕೂಡ ಈಕೋ ಬರ್ತಾ ಇದೆ.
ಹಲೋ ಸರ್.
ಹಲೋ,
ಹಲೋ,
ಹೇಳಿ ಹೇಳಿ ಹೇಳಿ. ಮಾತಾಡಿ.
ಕೇಳ್ತಾ ಇದ್ದೀರಾ?
ಹಲೋ,
ಹಲೋ ಸರ್.
ಹಲೋ,
ಹಾ ಸರ್.
ಹೇಳಿ ಹೇಳಿ. ಮಾತಾಡಿ. ಹಲೋ,
ಹಾ ಸರ್.
ಹೇಳಿ ಹೇಳಿ. ಶಂಕರಪ್ಪ, ನೀವು ಮಾತಾಡಬೇಡಿ, ಮ್ಯೂಟ್ ಮಾಡಿ ಎಲ್ಲಾರನು.
ನಾನು
ಹಲೋ
ಮಾಡಕ್ ಬರಲ್ಲ ಸರ್. ದೇವಪ್ಪ ಸರ್,
ಹೇಳಿ.
ನಾನ್ ಮೂಟ್ ಮಾಡಕ್ ಬರಲ್ಲ.
ಮಾತಾಡಬೇಡಿ, ಮ್ಯೂಟ್ ಮಾಡಿ ಎಲ್ಲಾರನು.
ನಾನ್ ಮಾಡಕ್ ಬರಲ್ಲ ಸರ್.
ಇರಿ ಇರಿ ಇರಿ, ಒಂದ್ ನಿಮಿಷ ಇರಿ ಶಂಕರಪ್ಪ, ನೀವು ಹೇಳಿ.
ನಾನ್ ಮೂಟ್ ಮಾಡಕ್ ಬರಲ್ಲ.
ಓಕೆ, ಆಯ್ತು, ನೀವು
ನಾನ್ ಮಾಡಕ್ ಬರಲ್ಲ.
ಪ್ರಶಾಂತ್
ಹಾ ಹೇಳಿ ಸರ್.
ಎಲ್ಲಾರ್ನು ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಡಿ, ಈಕೋ ಬರಬಾರ್ದು ಅಷ್ಟೇ. ಓಕೆ
ಒಂದ್ ಸೆಕೆಂಡ್ ಸರ್.
ಪ್ರಶಾಂತ್,
ಹಾ ಹೇಳಿ ಸರ್.
ಎಲ್ಲಾರ್ನು ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಡಿ, ಈಕೋ ಬರಬಾರ್ದು ಅಷ್ಟೇ. ಓಕೆ
ಒಂದ್ ಸೆಕೆಂಡ್ ಸರ್.
ಪ್ರಶಾಂತ್,
ಹಾ ಹೇಳಿ ಸರ್.
ಎಲ್ಲಾರ್ನು ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಡಿ, ಈಕೋ ಬರಬಾರ್ದು ಅಷ್ಟೇ. ಓಕೆ
ಒಂದ್ ಹಾ?
ಹಲೋ
ಹಲೋ.
ಪ್ರಶಾಂತ್,
ಹಾ ಹೇಳಿ ಸರ್.
ಈ ಕ್ಲಿಯರ್ ಆಗಿ ಕೇಳಿಸ್ತಾ ಇದೆಯಲ್ಲ?
ಹಾ ಕೇಳಿಸ್ತಾ ಇದೆ ಸರ್.
ಎಸ್, ಎಲ್ಲಾರ್ನು ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಡಿ.
ಹಾ ಎಲ್ಲಾರ್ನು ಮ್ಯೂಟ್ ಮಾಡ್ಬಿಟ್ಟಿದ್ದೀನಿ ಸರ್.
ನೀವು ಎಲ್ಲಿ ಹೋಗ್ಬೇಡಿ ಇವತ್ತು.
ಹೇಳಿ ಸರ್.
ನೀವು
ಹಾ ಕೇಳಿಸ್ತಾ ಇದೆ ಸರ್.
ಇದು ರಿಪೀಟ್ ಬರ್ತಾ ಇದೆ ಯಾಕೆ? ಸ್ವಲ್ಪ ದಿವಸ ಒಂದು 10 ಸೆಕೆಂಡ್ ಆದ್ಮೇಲೆ ಮತ್ತೆ ರಿಪೀಟ್ ಆಗ್ತಾ ಇದೆಯಲ್ಲ ಅದು? ಹಲೋ ಹಲೋ
ಹಲೋ.
ಹಾ ಹಲೋ,
ಹಾ ಸರ್.
ಕೇಳಿಸ್ತಾ ಇದೆಯಲ್ಲ?
ಹಾ ಕೇಳಿಸ್ತಾ ಇದೆ.
ಹಾ, ಈಗ ನಾವು ಮಾತಾಡಿದ ಮೇಲೆ
ಹಾ.
ಮತ್ತೆ ಸ್ವಲ್ಪ ಹೊತ್ತಿಗೆ ಈಕೋ ಆಗಬಾರ್ದು.
ಸರ್ ನನ್ನಲ್ಲಿ ಲೈವ್ ಚಾನಲ್ ಸ್ವಲ್ಪ ಆನ್ ಮಾಡಿದ್ದೆ.
ಹಾ,
ಅದಕ್ಕಾಗಿ ಅದ ಮತ್ತೆ ಅಲ್ಲಿ ಸ್ಟ್ರೀಮಿಂಗ್ ಆಗಿ ಅದ ಮತ್ತೆ ರಿಪೀಟ್ ಆಗ್ಲಾತಿತ್ತು.
ಓಕೆ ಓಕೆ, ಅದನ್ನ ಸಾಲ್ವ್ ಮಾಡ್ರಿ.
ಅದನ್ನ ಆಫ್ ಮಾಡಿಬಿಟ್ನೆ.
ಓಕೆ, ಡನ್ ಡನ್ ಡನ್.
ಸರ್ ನನ್ನಲ್ಲಿ ಲೈವ್ ಏನ್ ಬರ್ತೈತಿ ಬಂದ್ ಮಾಡಿಬಿಟ್ನೆ.
ಹಾ,
ಅದ ರಿಪೀಟ್ ಆಗೋದು ಬಂದ್ ಆಗಿಬಿಟ್ಟು.
ಓಕೆ, ನೌ ಇಟ್ ಇಸ್ ಓಕೆ ಅಲ್ಲ?
ಹಾ ಸರ್, ಹಾಸ್ಟ್ ಯಾರು ಮಾಡ್ಲಿ? ಹೋಸ್ಟ್
ಇರಿ ಒಂದ್ ನಿಮಿಷ. ಪ್ರವೀಣ್, ಹೋಸ್ಟ್ ಅಂಡ್ ಕೋ ಹೋಸ್ಟ್ ಯಾರು ಮಾಡ್ಬೇಕು? ಹಾ, ಒಂದು ಲಕ್ಷ್ಮೀ ಶಂಕರ್ ಅಂತ ಇದೆ ನೋಡಿ. ಮೆಸೇಜ್ ಮಾಡ್ತಾರೆ, ಮೆಸೇಜ್ ಮಾಡ್ತಾರೆ.
ಇಲ್ಲ ಅದಾರ ಸರ್.
ಹಾ, ಎಂಟ್ರಿ ಆಗ್ತಾರೆ.
ಒಂದಿಟ್ಟು ಚಾ ಕೊಡ್ರಿ ನಂಗ.
Change the name.
ప్లాట్ఫామ్ చేంజ్ చేసి సార్ ప్లాట్ఫామ్ చేంజ్ చేస్తే ఓకే సార్ థ్యాంక్ యూ సార్ థ్యాంక్ యూ కనిపెడతారు అంటే సార్ అక్కడ ప్యానలిస్టులో అందరూ కనిపెడతారు వీల్ బి వాచింగ్ సార్ ಅಂತ ವಿಡಿಯೋ ಇರ್ತಾವಂತ ವಿಡಿಯೋ ರೆಕಾರ್ಡಿಂಗ್ ಯೂಟ್ಯೂಬ್ ಲೈವ್ 1 second sir. 1 second. 1 second. 1 second. ಇಲ್ಲ ಬಿಡಿ ನಾನು ರೀಸೆಂಟ್ ಆಗಿ
ಇವಾಗ ಪರವಾಗಿಲ್ಲ ರಾತ್ರಿ ನಾನು ಹೋದ್ಮೇಲೆ ನಾನು ರಿಸರ್ಚ್ ಪಾರ್ಟಿ
ಅಲ್ಲ ಇದೇನು ಎಲ್ಲದ್ರಲ್ಲೂ ನಂದೇ ಕಾಣಿಸ್ತಿದೆ. ಇಲ್ಲ 2-3 ಕಡೆ ನಂದೇ ಬರ್ತಾ ಇದೆ. ಯಾಕೆ Good morning. Good morning,
Morning,
Chakita sir.
Good morning, Professor.
Very good morning.
This is excellent. Nice. Very clear.
So, chairman also here.
Yeah, yes, sir. I feel the presenters, Dinesh, Kumar, Dinesh.
Hello.
Hello, sir. Hello, sir.
ಹಲೋ ಹೇಳಿ ಪ್ರಶಾಂತ್.
ಹಾ ಸರ್, ಈಕೋ ಬಂದಾಯ್ತಲ್ಲ?
ಓಕೆ, ಈಗೋ ಬರ್ತಾ ಇಲ್ಲ ಈಗ.
ಹಾ ಓಕೆ ಸರ್, ನಿಮ್ಮದಲ್ಲಿ ಯೂಟ್ಯೂಬ್ ಚಾನಲ್ ಏನ್ರಿ ಸ್ಟಾರ್ಟ್ ಇದ್ರೆ ಅದ ಈಕೋ ಆಗ್ತದೆ, ಅದಿದ್ರೆ ಬಂದ್ ಮಾಡಿ.
ಈಗೋ ಯೂಟ್ಯೂಬ್ ಏನ್ ಮಾಡ್ಬೇಕು?
ಸರ್ ನಿಮ್ಮಲ್ಲಿ ಹತ್ರ ಎಲ್ಲೇ ಮತ್ತೆ ಯೂಟ್ಯೂಬ್ ಚಾನಲ್ ನೀವು ನೋಡ್ತಾ ಇದ್ರ ಅಂದ್ರ ಅದು ಈಕೋ ಆಗಿಬಿಡ್ತೈತಿ.
ಎಲ್ಲ ನಮ್ಮಿದ್ರಲ್ಲ
ಹಾ, ನಿಮ್ಮ ಕಂಪ್ಯೂಟರ್ದಲ್ಲಿ ಮತ್ತೆ ನೀವು ಯೂಟ್ಯೂಬ್ ಚಾನಲ್ ಚಾಲು ಮಾಡಿದ್ರಿ ಲೈವ್.
ನೋಡಿ, ಅಲ್ಲಿ ಲೈವ್ ಅಂತಿದೆ ನೋಡಿ.
ಲೈವ್ ಅಲ್ಲಿ ಸ್ಟಾರ್ಟ್ ಇಲ್ಲ ಸರ್ ನಿಮ್ಮಲ್ಲಿ?
ಲೈವ್ ಇಲ್ಲ.
ಹಾ, ಲೈವ್ ಇಲ್ಲ ಅಂದ್ರೆ ಈಕೋ ಆಗೋದಿಲ್ಲ ಇನ್ನ.
ಇಲ್ಲ, ಲೈವ್ ಇಲ್ಲ, ನನ್ನ ಹತ್ರ ಲೈವ್ ಇಲ್ಲ.
ಓಕೆ ಸರ್, ಈಗ ಅದಕ್ಕೆ ಈಕೋ ಆಗ್ತಾ ಇಲ್ಲ. ಆಗಲೇ ಮಾತಾಡ್ರಲ್ಲ, ಈಕೋ ಆಗಿ ಬಿಡ್ತಾರ.
ಓಕೆ, ನಾವು ದೇರ್ ಇಸ್ ನೋ ಲೈವ್. ನನ್ನ ಹತ್ರ ಲೈವ್ ಮಾಡಿಲ್ಲ.
ನಿಮ್ಗ ಸರ್ ಪೋಸ್ಟ್ ಮಾಡ್ಲಿ?
ಇಲ್ಲ ಇಲ್ಲ, ನಂದು ಬೇಡ. ಡಿಜಿಫೆಕ್ಸ್ ಅಂತ ಇತ್ತಲ್ಲ,
ಡಿಕೆ ಪಿಕ್ಸ್ ಇನ್ನ ಬಂದಿಲ್ಲ ಸರ್ ಇಲ್ಲಿ.
ಇರಿ ಕೋವೋಸ್ಟ್ ಯಾರು ಮಾಡ್ಬೇಕಾದ್ರೆ ಕೇಳಿ ಪ್ರಶಾಂತ್. ಲಕ್ಷ್ಮೀ ಶಂಕರ್ ಅಂತಿದೆ ನೋಡಿ.
ಹಾ, ಲಕ್ಷ್ಮೀ ಶಂಕರ್ ಕೋ ಹೋಸ್ಟ್ ಮಾಡ್ಲಿ ಸರ್? ಹಾ, ಕೋ ಹೋಸ್ಟ್.
ಎಸ್ ಮಾಡಿ. ಶಂಕರಪ್ಪ ಸುಬ್ಬಣ್ಣ ಎರಡಿದಾವ್ ನೋಡಿ.
ಯಾವ್ದು ಸರ್?
ಲಕ್ಷ್ಮೀ ಶಂಕರ್ ಒಂದು, ಶಂಕರಪ್ಪ ಸುಬ್ಬಣ್ಣ ಅಂತಿದೆ ನೋಡಿ.
ಹಾ ಹಾ,
ಎರಡು ಮಾಡಿ, ಎಲ್ಲ ಮಾಡಿಬಿಡಿ.
ಹಾ ಓಕೆ ಸರ್.
ಮ್ಯೂಟ್ ಮಾಡಿದ್ರಲ್ಲ?
ಹಾ, ಎಲ್ಲಾರು ಮಾಡಿ ಸರ್. ಅವರು ಇನ್ ಮಾಡ್ಬೋದು, ಮ್ಯೂಟ ಅನ್ಮ್ಯೂಟ್ ಅದು ಎಲ್ಲ.
ಓಕೆ.
Okay, sir.
ಪ್ರೊಫೆಸರ್ ಶ್ಯಾಮರಾವ್.
Professor, good morning.
ವೆರಿ ಗುಡ್. ವೆರಿ ಗುಡ್.
I feel speakers have also joined.
During time, we recommend open the applications or other content that you want to share before you join the meeting. To start in a meeting, go to meeting controls and click share content. You can optimize the font based on whether you share documents and images or video and multimedia. Now, choose what you want to share. The easiest way to share content in the meeting is by sharing your screen. Select which screen you want to share. You can see everything open up your desktop. If you want more control, that you share an application instead of a screen. This gives you the security of sharing an application without private content, such as an email. When sharing a specific application, meeting participants only see the application that you choose. Keeping your desktop and any other applications private, participants won't be able to see your other windows, even if they're on top. Let me show you a few different ways to share content during a Cisco WebEx meeting. To save time, I recommend that you open the applications or other content that you want to share before you join the meeting. To start sharing content in the meeting, go to the meeting controls and click Share Content. You can optimize the quality based on whether you plan to share documents and images.
ಡಾಕ್ಟರ್ ಸಂಗೀತಾ, ಹಲೋ ಹಲೋ.
ಕೇಳಿಸ್ತಲ್ಲ ನಂಗೆ ಏಕೆ ಸಿಕ್ಕದೆ ಅದು ಡಿಸ್ಕನೆಕ್ಟ್ ಮಾಡಿದೆ ಎಲ್ಲೋ ಬರ್ತಾ ಇದೆಯಾ
ನೀವು ಮಾತಾಡ್ಬೇಕು ಮ್ಯೂಟ್ ಮಾಡ್ಬೇಕು.
ಗುಡ್ ಮಾರ್ನಿಂಗ್ ದೇವಪ್ಪ ಸರ್ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ
ಕೇಳಿಸ್ತಾ ಇದೆ. ಪ್ರೊಫೆಸರ್ ದಿನೇಶ್ has entered.
Yes, yes.
Okay, no problem, sir. Please take your own time.
Now, do you know how to do it from Mac?
Sir, there is one.
Just beside stop video, there is a share content.
No. This is the three options are there in the bottom of the computer. Mute, stop video, and share.
Yeah.
Click on the share mark.
It says how do you do it?
Sir?
See, there is nothing like
Sir, can I help? Sir, in the bottom of the
when
computer.
I go to the share, there is no green to click on it. So,
No, there is no need of green. The black itself, it will work. No problem.
okay.
You can click on that.
Yeah. It tells me open system preferences.
Yes, sir. So in the beginning, sir, please open the PPT on the desktop.
Okay. Yeah. Yeah. So it's open actually. Yeah.
Oh, okay. Then you click on the share button. That's all. It's very simple, sir.
No. No. Okay. Yeah.
Share content, sir.
Yep.
Yeah, you're sharing your meeting. So close that.
Which one? Close.
Close.
This one.
Yeah.
Okay. And then
Just close. Open that.
yeah.
Yeah.
Yeah. Okay.
Open that up. Yep. Yeah, let's see.
Do you say screen or?
Sir, viewing, sir. It is viewing, sir.
Okay.
Okay.
Neha?
Hello, sir.
Can you see my screen?
Yeah,
Not
yeah.
yet, sir.
Yeah. Okay.
Yes.
Share ಮಾಡಕ್ಕೆ business is a problem. Can you share it? It's not ours. Okay. Ours is.
Sir, Dinesh sir, we are seeing you. We are able to see your screen, not your presentation slide or whatever. It is just your screen with all the participants.
You see, I can go into the. Let's see. Now?
It says viewing Dinesh's meeting.
Uh,
Just a minute. You are clicking something YouTube. Share my meeting window. Yeah.
But now?
No. Sir, maybe you can open your presentation, and here there is a share button. I think you need to click that. Can you help us?
Hello, sir.
Sir, our technical person is here, sir.
Hello.
Yeah.
Sir, kindly keep your PPT open.
It's open. Yeah.
Now go to the share.
Yeah.
Here on the.
Click on the share button. Can you see your PPT over there?
What are you trying?
Can you click on a?
We're not seeing it anymore.
Oh, what?
I have to go here. No.
Hello.
Hello?
So when you click on share, you have to see your PPT over there.
Uh,
You have to select the PPT.
No, sir, not.
Not the share meeting window.
yeah.
Yeah, disable that, sir.
It is
Unclick that.
yeah. Okay. Yeah.
Sir, on your screen, you can see mute button, stop video button. Next to that, the share button.
Yeah, that share button doesn't seem to work.
Yeah, click on the share. See to that. You have to find the PPT in that particular.
Open system preferences.
Really?
Then come back at work.
No.
Yeah.
They won't. No. Oh, you mean turn off?
Leave and come back.
Here?
No, you leave and then,
Turn off?
yeah,
And then
then come back.
quit?
Yeah, can't you just come back again?
Yeah.
Yes, sir. Yes, sir.
Yes, sir, it is coming.
Yes, sir. Sir, make a full view, sir.
Yes, yes, sir. Dinesh, sir, we can see your this one PPT now.
Okay.
Good. That. Yes, sir. Yes, sir.
You can tap. No, you can tap to share. No. ಇದು ನಿಮ್ಮ ವಿಡಿಯೋ
ಹಾರ್ಡ್ ಮಾಡಿ ಅವರೇ ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಮೂಟ್ ಅಷ್ಟೇ. Okay.
Very good morning to one and all. I welcome you for the day 2 of the international conference. I first extend a warm welcome to our distinguished chairman, co-chairman, and speakers for microbial interactions and signaling. Now, first of all, I would like to welcome the chairman for this session, Dr. Tirtha Prasad sir, who was the Dean of Agriculture, Hassan, and was a professor and head of Biotechnology Department, UAS Bangalore, who has more than 120 international papers to his credit and has guided about 28 PhD students and 50 MSc students. Welcome to this session, sir. Next, I would also like to thank you, sir.
Thank you.
I would like to welcome Dr. Shamrao Jagirdar, Professor of Plant Pathology, University of Agriculture Sciences, Dharwad, who has about 175 papers and is a life member for more than 20 societies, who has also received many awards to his credit. And now we have very distinguished speakers for this session, Dr. Professor Dinesh Kumar, who's going to speak on emerging plant immunity, who is from the University of California, Davis, and we have Jagdish Gupta, who's going to talk about the role of nitrogen nutrition and nitric oxide signaling in plant pathogen interactions. I extend a warm welcome to all of you, sir. Now over to the chairman and co-chairman.
Hello.
Welcome to speakers, co-speakers of this particular session. Dr. Dinesh, I am Tirtha Prasad here. Hello. Dinesh, I am Tirtha Prasad here, the chairman of the session. Can you hear me, Dinesh?
Yes, to unmute, sir. Yes, sir, unmute yourself and then.
I'm actually unmuted. Can you hear me?
Yes.
Sir. Yes, sir. We can hear you.
Now Dr. Shamrav is going to introduce Dr.
Thank you, sir. Good morning to everyone. I welcome all for the second day's international conference on the multidisciplinary approaches for sustainable management of crop diseases. Yesterday, we had a very good session with leading up by the DTG ICAR inaugurated, and we had a very fruitful session. Sir, I have the privilege to introduce the keynote speaker. Today's keynote speaker, Professor Dinesh Kumar, SP. He is a professor of plant biology and chair at the Department of Plant Biology, University of California, Davis. Professor Dinesh Kumar, sir, is an internationally accomplished scientist having significant contributions in the plant molecular microbial interaction.
Shamrav, sir, you have muted yourself, sir.
Madam, now it is okay?
Yes. Yes, now it is okay, sir.
Right, madam. Then once again, I have the privilege to introduce Professor SP Dinesh Kumar, sir. He is a professor of plant biology and chair of at the Department of Plant Biology, University of California, Davis campus. Professor Dinesh Kumar is an internationally recognized scientist for his significant contributions on molecular basis of host pathogen interaction. He has contributed a lot and opened a new window of opportunity for the science, especially with respect to the chloroplast function and autophagy. He and his colleagues have developed an understanding of the role of chloroplast in plant pathogen recognition and defense system. Professor Dinesh Kumar has developed tools which are being internationally accomplished, especially with respect to gene function studies, including the tobacco rattle virus. And these have specific, they have the
Shamrav, sir, you have muted, sir.
it's muting. Madam, somebody is muting. Somebody is muting, I don't know. And viral system to deliver the gDNA is
Yes, sir.
especially using the CRISPR-Cas9 technology, which is the latest tool in the host pathogen interaction. Professor Dinesh Kumar has discoveries led to creation of new paradigms with respect to and answered many crucial answers with respect to the host pathogen interaction, especially the pathogen triggered immunity and the PAMs and MAMs, etc. He has more than 110 research papers to his credit with high impact factor journal publications, 11 books chapters, and he holds 3 patents. His research accomplishments have been recognized by the world society, especially he is a fellow of the American Association for Advancement of Science in the year 2018, and he is the Davis Faculty Research Award in 2016, Junior Faculty Research Award in 2003, and Hallman Family Fellowship in 2000. He is serving as editor of many of the internationally accomplished journals. To quote a few, he is a senior editor of MPMI, Plant Molecular Microbial Interactions, from 2004 to 2006. He is an advisory member of Plant Journal from 2004 to 2006, and he is editorial board member of Autophagy from 2006 to 2010. Currently, he is a member editor of an well-recognized high-impact factor journal Plus One Pathogens, a senior editor of phytopathology research, editorial board member of molecular and cellular proteomics, and an editor of the PIRJ. He is a member of American Society of Plant Biology, Minority Affairs Committee. Dinesh Kumar has trained numerous graduates, postgraduates, postdoctoral researchers in his laboratory throughout the world. The students, the faculty visited his lab and learned a lot with respect to the host pathogen interaction. So I invite on behalf of the organizing committee, on my personal behalf, for having accepted our invitation to give his keynote address on emerging perspectives on plant immunity. Professor Dinesh Kumar, sir, you have the allotted time of the 30 minutes. After 25 minutes, I will show a sign of 5 minutes left, and you have to conclude, and we will have 2 minutes discussion. Now the floor is yours, sir.
Okay, so can you hear me?
Yes, sir, yes.
Okay, so I like to thank Dr. Shama there for a very kind introduction, actually. I also want to thank Dr. Devappa and Sangeeta for inviting me to participate in this exciting e-conference. So my lab has been working on plant immunity in the last 20 years or so. I'm going to tell you a little bit about what we do in the next 24 minutes, actually. So let me see if I can. Okay. As you all know, as this conference title indicates, the impact of plant pathogens is very high, and hence many of us here study on how actually pathogens cause diseases in plants, actually. So classic example is like Irish potato famine, which caused a major issue. Millions of people migrated out of Ireland into the United States and other parts of the world because of the loss of the potato crop due to the oomycete pathogen Phytophthora infestans, actually. And then recently in 1999, this strain of the wheat rust called UG99 is creating an issue. Initially started out in Uganda, and now it's moved to other parts of the world, including India, where it's creating major issues, actually. And then in the United States and in Asia, citrus greening is becoming another problem. Estimate that if we don't have some solution for this disease in the next 20 years, we may not have oranges to eat or drink oranges, actually. So, and finally, I want to clear is that in sub-Saharan Africa, for example, viral diseases are major problems. And in cassava, especially the cassava mosaic Gemini viruses have major issues. I know there are some speakers here will be talking about these Gemini virus issues, actually. So there is an annual crop loss about 1.9 to 2.7, actually. So all these incidences illustrates why we need to study how plants actually defend against pathogens and how pathogens actually causes diseases, actually. But having said that, the disease is not a common process in plants. Most of the plants actually can resist the microbial pathogens, actually. So that's the reason why we see a lot of healthy plants, outside of our home or outside of the buildings, actually. But of course, the established pathogens, given a proper condition, can actually cause major issues, especially in the crop plants. So the reason plants can actually overcome the infection is because they have this sophisticated immune systems. Although plants are not able to move like humans, but they do have an immune system which can actually function similar to how the human immune system can function, even though the plants don't have these specialized immune cells like neutrophils or macrophages, actually. So that's because they contain this large number of immune receptors, which functions to defend against pathogens, actually. So just to give you an idea here, a very simplistic way of the immune system is that the plants have these cell surface receptors, and these receptors generally recognize general molecules of pathogen, like, for example, flagella of the bacteria or chitin of the fungal pathogen, and activate an immune response with several processes which I'll come back to that soon, activating gene expression and proteome changes, which ultimately lead to the containment of the pathogen, actually. But of course, pathogens are not naive to take this offense. So they actually evolved to inject proteins into the host cells. And these proteins are often referred to as effectors, and these effectors interfere with this pathway here, actually. So hence, the plants have evolved these another kind of intracellular receptors, and these are often referred to as NLRs. And these actually recognize these effectors and activate an immune response to contain the pathogen to the infection site, actually. So although this is a simplistic view, but pretty much we know that this is how part of the immune system works in plants, actually. Okay. So, but what is also interesting is that this conceptual framework seems to be conserved across different organisms, actually. So what I alluded to just now is these receptors can recognize PAMs plants, receptor kinases. Similarly, in Drosophila, these receptors, which have similar domains, can actually recognize pathogens. And in the humans, there are about 11 toll-like receptors which can actually recognize pathogen, which contain these common structural domains which have evolved to recognize pathogens, actually. And in addition, I alluded in plants, they have these intracellular receptors to overcome pathogen infection. Similarly, in the mammalian system, similar molecules exist to recognize pathogens, actually. So what this suggests is that the system seemed to have evolved to contain these structural domains which seem to recognize pathogen to activate immune response in different organisms, actually. So over the years, we have been interested in how these receptors, like pattern recognition receptors in the cell surface, and how these intracellular receptors actually recognize pathogens and activate an immune response. Actually, I'm just listing you papers here if you're interested in looking at them. The other area of interest, as Dr. Shamarao indicated in recent years, there seems to be communication between chloroplast nucleus and other organelles during immune response in plants. So we are also interested in how these chloroplasts actually make these long tubular structures connecting the nucleus, and what are they actually transporting to the nucleus, and what is the significance of this connection in terms of activating a defense response in plants, actually. So although I don't have time to talk about this topic here, what I'll tell you a little bit about is that although we know these receptors since last 20 years, for example, the first NLR was identified by me and my colleague when I was in Barbara Baker's lab as a postdoc in UC Berkeley, but still 20 years later, we still don't understand how these proteins function and what these proteins interact with. So that is primarily because these proteins are expressed at very low level, and it is hard to identify proteins that interact with these proteins, actually. So recently, in the last couple of years, what we decided to do is to explore this option of the technique called proximity labeling, which is being optimized quite a bit in mammalian system, actually. So although it is optimized in the mammalian system, when we initially started to work with this approach, it's optimal in plants. So we decided to re-optimize this system. So what I'm telling you here is that dropping you a lot of background, but you can read the paper recently published. I will show you some data here. So we are using this biotin ligase called turbo-ID or mini turbo-ID, actually. So this is a modified version of an E. coli biotin ligase. So what it does is that when you put this biotin ligase to bake protein of your interest, in our case, immune receptor, so when you express that in the cell, this biotin ligase promiscuously labels all the lysins with biotin. It incubate the tissue or the cell with biotin anywhere from 3 to 24 hours at room temperature, actually. So when it labels the biotin, the proteins around and also on its own protein, actually. So simply what you can do is you can bind these biotin to the streptavidin beads and purify your interactors and identify what those labeled proteins are, actually. So the advantage here is that once you label it, when you're purifying, even if they fall apart, it's okay because it's already labeled, actually. So this is a major advantage over the technique that you use purification, where complex need to be kept intact until the last step, actually. Whereas in this approach, since you already labeled inside the cell, you don't care whether the complex falls apart, actually. So basically, the visiting scientist in the group, Yang Liang Zhang, a couple of years ago when he came, was able to optimize the system using different iodine ligases, BioID, BioID2, and TurboID, actually. So what we showed in this paper, which was published recently in Nature Communications, is that the TurboID is actually, whether it's at room temperature or 37 degree, performs much better in terms of labeling proximal proteins than the BioID or BioID2. So the bottom line conclusion of a lot of the data which I'm seeing is that a TurboID binding ligase performs better plants than other ligases. So then Young with others went on to purify complexes of the immune receptor using this approach. So the immune receptor we are interested in is receptor called N, which confers resistance to tobacco mosaic virus and other tobacco viruses in solanacea, like tomato, actually. So to cut the lot of the details chart here, what we have done here is using proximity labeling, combining with what is referred to as TMT labeling and quantitative proteomics with lots of different biological replicates, which is shown here. We've been able to identify proteins that interact with this immune receptor, actually. So then we took about 10 proteins that interact with the immune receptor and confirmed in vivo that they do actually interact. And then we begin to analyze their function in vivo. So I'm just showing you the snapshot of that result here. So we silenced using tobacco virus based Wix vector that Dr. Shamarab indicated in the introduction, and analyzed the function of these proteins, actually. So as you can see here, the control plant is completely resistant to the tobacco mosaic virus, so it is completely healthy, actually. But if you silence N itself, so obviously it loses resistance to TMV, and you can see it completely loses resistance, actually. But then if you silence these NLRs, you also lose resistance. This is really interesting because N itself is an NLR, but it also interacts with other NLRs, actually. So what it says is that these NLR receptors in plants work as a network of proteins immune response. Hello? And other thing you can see here is that these NLRs seem to function as a enhanced resistance, actually. So when you silence these NLRs, you get enhanced resistance pathogen, actually. So what it says is that these NLRs functions as negative regulator. So it seems like NLR network involved, some of them function as positive regulator and some of them function as negative regulators, actually. So one of the protein we followed up to understand the mechanistic basis of the function is this protein called UPR7, which belongs to a novel E3 ligase protein degradation machinery component, actually. So the reason we follow up on this is that the EPR family of E3 ligases is understudied, not just in plants, but also in the other organisms. So there are 7 UPR proteins, and most of the function of these proteins is unknown. Interestingly enough, UBR2 and 4 also functions in immunity in the human system, actually. So consistent with UBR7 being an E3 ligase, what happens really is that it seems to regulate the receptor level, actually. So if you overexpress the UBR7, you can see you lose the protein amount, actually, compared to the control here. But if you treat with the proteasome inhibitor now, you completely rescue the amount of the protein, suggesting that this E3 ligase functions through the proteasome, which was not done before, actually. And consistent with that, if you silence the UBR7, you increase the protein amount. But if you treat with MG132, you also increase the amount, suggesting that UBR7, in fact, works through the MG132, actually. So basically, based on all the other data published in this paper, what we propose is that since immune receptor function is to activate immunity, part of the immunity is to kill the cells that are being infected, actually. So you don't want to have too much of this immune receptor in the cell. Otherwise, you will create an autoimmune response, so which is what the problem in the human inflammasome, actually. So what UBR7 is doing is that they're keeping the immune receptor content low, but when the virus comes in, it actually disrupts this interaction between the immune receptor and the E3 ligase. And now the immune receptor becomes more stable and hence activate the immune response when needed. So this is how we speculate that this E3 ligase is working, and we have a lot more work to do to understand the precise function of UBR7, not just in this pathway, but also in the other immunity pathways.